So over the next 10 minutes, what I would like to do is to...
Can you hold it close, Dr.
Yes, sorry, yeah. So talking to physicians and patients around the country, I realized that in fact there are probably a few distinct clinical phenotypes of the lung disease in systemic juvenile idiopathic arthritis. And what I would like to do over the next 10 minutes is to go over the phenotype that we are discussing here today. Certainly, if you sort of go back 10, 15 years ago, the lung disease in systemic JI, it's not something that we saw very, very frequently. In fact, the parenchymal lung disease was exceptionally rare, and perhaps the best description of this parenchymal lung disease was provided by Balua Thrier in 1980, and he described very mild, non-specific inflammatory infiltration in the interstitium, and he stressed the point that that inflammatory activity, in fact, did not have a major impact on the lung function and the quality of life of those patients. It was actually in 2012 when Yuki realized that, in fact, we might be dealing with a new emerging pattern of the lung disease associated with very high mortality rate. And she, in fact, was the first one who started using the term alveolar proteinosis in our literature. And unfortunately, the Cincinnati experience happened to be very, very similar. Our first patient with this type of lung disease was diagnosed in 2010, and then we had a major uptick in number of those patients since 2012, and again, conspicuously did coincide with the introduction of biologics. So as of 2018, the total number of patients with this lung disease in Cincinnati was 18. Grant, correct me if wrong, as of last week, it's 27, right? So this is certainly is very, very concerning. Now, when we looked at, and let's focus on those 19 patients that we diagnosed between 2010 and 2018. So as a group, these patients tend to be younger. I would say, at least in our cohort, at least half of them, they developed the first signs of the disease before their second birthday. All these kids had prominent systemic features, and I would say persistent systemic features. It's very different from the rest of the kids with SJIA, where systemic features, and whom systemic features tend to go away over time. The arthritis is not very prominent, and if it is present, usually it's just a few large joints, often affected in a rather asymmetric manner, I would say. At least 80% of patients in our cohort had the history of macrophage activation syndrome, usually recurrent. And then certainly this is very striking as well. Almost 40% of the patients in our cohort had this bizarre anaphylaxis-like reaction to tocilizumab. And now actually we had another patient who had a similar reaction to canakinumab as well. And then...
We see this in Candle too, I don't know if you know. So I mean...
Mm-hmm. Mm-hmm.
Can you confirm? You mentioned it, but I know I've heard it. Sub-Q is fine, IV is not, so it's not the molecule itself. It's something else. It's something else which is in the body.
Yeah, and it's not an allergic reaction.
No, I know. We have discussed this last week.
The skin lesions. I mean, for us, it was not skin lesions. He was just really, really sick, and that was the only time he was hospitalized without, and the inflammatory lab became normal. They were fine. And it was the day after he got the first, infusion of tocilizumab.
But now I had one patient who had a similar reaction to canakinumab as well, and she's actually developing lung disease right this moment. So I think it's not limited to just tocilizumab. Yeah, that's correct. 11, yeah. Mm-hmm.
Am I doing the math right?
9 plus 2, yes.
All of them got at least 8 infusion. It's several infusions.
Yeah.
It's curious, right? It's not the molecule.
Could it be a preservative? I mean, what else is in those?
Secret sauce. Cell activated.
What I heard was I have two patients with polyarticular JIA who have infusion reaction with tocilizumab and who do well once you give them subcutaneously.
Mm-hmm.
So it may not be necessarily associated with the disease.
But Hermina, I mean, the proportion of patients developing this reaction in this particular group of patients is striking. It's more than 40%, Grant, right?
In the clinical trial, it was like 1%.
Yeah, yeah. Mm-hmm.
Intended
Right.
for-
In the systemic.
Mm-hmm.
But is that something,
SJIA path?
the difference between IV and reactions and sub-Q, is that something that has been seen in general? Do you know? I guess we can ask Rosa and see, we're gonna look at the safety report, it's not complicated.
Yeah, I think,
It also happens in asthma. So with Cellular, you can get anaphylactic reactions, and the antibodies against IL-5 that are given IV can cause that, but not when given sub-Q. So I think it's more of a general phenomenon rather than a
Specific.
problem
Mhm.
associated with our patients.
Still, there is something goofy about this.
There's no doubt it's real. Is it a drug or is it?
So the next point I would like to make is that we totally agree with Raphael. The levels of IL-18 in these patients actually are striking. So to me right now, if I see a new patient with systemic juvenile idiopathic arthritis, developing the disease early in life with prominent systemic features, kind of high ferritin, propensity to develop MAS, high level of IL-18, I know that I need to watch this kid. I think this kid may develop macrophage activation syndrome. Now, most of these patients who came to Cincinnati, we saw in Cincinnati, came to us for a second opinion. But when sort of we look at the zip codes, we sort of realize that at least 6 patients, now not 5, 6 patients were clearly local patients coming from our catch area. And on a continuous basis, we are following somewhere between 75 and 100% of patients with systemic juvenile idiopathic arthritis. So if our numbers are correct, then the prevalence of this phenomenon could be as high as 6% and probably even actually higher than this particular pattern that I'm describing here. So all of the kids that we have in that cohort had this particular pattern. And sort of the other striking feature, and all the parents today, Sophia, Veda, all the parents mentioned the fact that there was a striking dissociation between the sort of relatively mild clinical presentation, some clubbing, perhaps a little bit of tachypnea with physical activities, and then the chest CT findings were very, very dramatic. And again, sort of I'm showing you the spectrum of those findings. So typically in a mild case, we would see a little bit of pleural thickening, maybe a little bit of the tree and bad passages. But then sort of we see more pleural thickening, interceptal thickening as well. And then in at least half of the cases in our cohort, we saw that sort of massive ground glass-like passages consistent with interstitial process. And again, many patients would develop areas of consolidation. Now, we performed open biopsies in Cincinnati in 6 patients, and also we got additional biopsies from other places for our review. And the findings actually are quite striking. So typically we see patchy, but often very extensive lesions comprised of mixed features of pulmonary alveolar proteinosis and endogenous lipoid pneumonia. We also see this sort of pleural and interlobal septal fibrosis, and in many cases we saw also prominent vasculopathy, and the extensive infectious workup in those biopsies was negative. And I would like to show you some examples. So that was actually a kid who came to us for a second opinion, absolutely no lung symptoms, pulmonary symptoms, but we did notice a little bit of clubbing. He was one of the first in that cohort, so we actually did decide to proceed with a lung biopsy, and that's sort of how it looked. That's a gross appearance of the lung, right? And I would like to draw your attention to a couple of things. So there are very healthy looking areas of the lungs, and then there are these sort of cheesy looking areas. And if you look at that particular piece of tissue under at H&E staining, you see sort of patchy but very extensive infiltration. Now, if you look at these areas at a higher level of magnification, you see a couple of things. First, you see this alveolar filling with cholesterol clefts and this proteinaceous material. Then the other sort of very prominent feature is sort of massive interstitial infiltration. And in some areas, it was really, truly striking. The next thing I would like to draw your attention to is the fact that the vasculopathy was prominent in many cases. In this example, we are looking at the venule. Here it's an artery, and you see sort of significant thickening of the muscle layer of the blood vessel. Interestingly, of those 5 patients who had this pattern in our cohort, I believe only one had pulmonary hypertension based on echo. So again, sort of, it could be very dramatic, but for whatever reason, clinically, it may not be that apparent. And the other point I would like to make here is that this process eventually leads to significant fibrosis. And in this example, we are looking at the pleural sort of septal fibrosis that is certainly very, very prominent. Now, we also looked a little bit at the composition of the cellular infiltrate in the lung tissues. Here I'm showing you T-cell staining, B-cell staining, certainly some areas of mild accumulation of B cells, but certainly we see a marked predominance of T cells. So if you look at the T cells in general, I would say CD4 cells predominate, although now we had 2 patients in whom we saw equal or maybe even slight predominance of CD8 cells. And I'm not showing it here, but we also did staining for a couple of T cell activation markers, and these T cells clearly look very activated. Now, coming back to this sort of idea of pulmonary alveolar proteinosis, certainly this alveolar filling with this proteinaceous material with cholesterol clefts is awfully reminiscent of what might be seen in primary PAP. And if you sort of look at the literature of primary PAP, typically in these patients we may see 2 things. We may see surfactant dysfunction secondary to mutations in the gene encoding surfactant, or we can see macrophage dysfunction that is secondary to impaired GM-CSF signaling. And that could happen for 2 reasons. We either have mutations in the GM-CSF receptor, or these patients develop neutralizing anti-GM-CSF autoantibodies. Now, and the point is that sort of it looks like in both autoimmune and genetic PAP, loss of this signaling impairs the ability of alveolar macrophages to catabolize surfactant. I would like to explain this a little bit better. So here we're looking at the alveolar macrophages. So typically there is a population of those macrophages that is supposed to catabolize or recycle surfactant. So these macrophages phagocytose surfactant, and then that little phagosome is supposed to fuse with a lysosome. And that fusion eventually leads to a complete degradation of surfactant, and everything returns back to normal. Now, the interesting thing is that if we don't have proper GM-CSF signaling, for whatever reason, whether it's a genetic mutation or neutralizing anti-GM-CSF antibody, we see that these macrophages still try to phagocytose surfactant, but we don't see the fusion between the phagosome and the lysosome. And the result, we see accumulation of these phagosomes with unrecycled, uncatabolized surfactant in the cytoplasm of a macrophage. And that leads to 2 things. It leads to the emergence of those foamy macrophages filled with those what we call lamellar bodies, those phagosomes with unrecycled surfactant. And then we sort of see this milky substance filling the alveolar space. So when we certainly saw similar features in our patient, and that's Vikram's electron micrograph of his lung. Here we are looking at his alveolar macrophage at a very high level of magnification, and what I would like sort of you to see is those lamellar bodies, right? Those phagosomes that are filled with this unrecycled, uncatabolized surfactant. This is certainly strikingly similar to genetic PAP, so when we realized that, we immediately performed whole exome sequencing and whole genome sequencing as well. Well, now I think we have more than 10 patients now, and I think you have the data, right? The sequence data. I believe we sent it to you. Yeah, and the point is that sort of we tried hard. We looked at all the genes that had been associated with primary forms, genetic forms of PAP, and we could not find even a single pathogenic variant. We also did some additional sort of genome-wide search, did not find much, but I think you may have some ideas what it might be, correct?
We have something we're pursuing, but it's not monogenic, I can tell you. I think
Right.
it's actually, it's probably a tuning process.
Mm-hmm. So, but the point is that sort of looking at least at the genes, and it's a long list, it's about 20 different genes associated with primary PEP, not a single pathogenic variant. We also looked at the presence of neutralizing anti-GM-CSF antibodies in 6 patients with this phenotype, and there was absolutely nothing. So at the moment, we don't think it's genetic. We don't think it's autoimmune PEP. We also looked a little bit at the signaling pathway itself. So again, remember, to stimulate that fusion between the phagosome and the lysosome, we need GM-CSF signaling, and GM-CSF signals through STAT5. So we took samples, monocytes, fresh monocytes from, I believe, 5 patients with systemic JIA in this particular lung disease. We put those monocytes in culture and stimulated them with different concentrations of GM-CSF. And as you can see, sort of we see beautiful, beautiful signaling. So those are peripheral blood monocytes. So clearly the signaling pathway, at least in the periphery, is not affected. So this is not a negative control. That's sort of an interesting sample. That's, in fact, those are those foamy macrophages from the BAL. So we did the same thing with those macrophages, and certainly there is some signaling, right? But it's not as impressive as in peripheral monocytes. So we don't know whether it's truly a reflection of decreased signaling or it's a sort of a technical artifact, because for this particular experiment, we could not come up with a decent negative and positive control, right? So imagine, so you want your antibody to get to that phosphorylated stat, right, to be able to pick up signaling. So the question is, when the macrophage is filled with those phagosomes, right, it may be technically difficult for the antibody to get to the right place.
I think you have to look at surface receptors, because remember, these are, I'm not sure if they are M1s or M2s, but they are macrophages, and they change their receptor on the surface. So, I mean, GM-CSF receptors are expressed on neutrophils, but they actually are differentially regulated on M2 and on M2 macrophages. So I think I would stain them and figure out what surface receptors do they actually have at that state, because they are strangely differentiated. And then also basically look at alveolar macrophages from any other sample you can get your hands on and see and stain the surface receptors and see has that actually changed. Because I do, as I said, I think there's a differentiation problem and you may not even have the receptor. So I don't think that that's abnormal, because you're comparing monocytes versus to macrophages. And M2 macrophages have actually been stimulated with MCSF in order to get to this point and various other things. So they very likely have downregulated their well, at least some of the MCSF receptor may actually be downregulated. It's a dimer. So look at the surface.
Yeah, the other control that you can do is actually take similar macrophages and stimulate the other side of kind of around phosphostatin.
That's true. Just use
Yes, I think that's,
something they express the receptor
Right,
to.
exactly.
Just but
Yeah,
phenotype
that-
them because they are weird.
yeah, go ahead, yes. And Alex, we need to look also at that, at the gene expression data as well, so it potentially can, we have single cell analytic data, so we might explore some of it in that data set. So anyway, the point I'm trying to make is that so far, you know, we've tried hard. We have not been able to identify any intrinsic issue with the macrophages. So the question is, are we dealing with some kind of extrinsic influence on these macrophages? And since we see it predominantly, almost exclusively in systemic JIA, so is it the inflammatory milieu of the underlying disease? The other thing I would like to mention is that sort of, you know, we tend to call this disease PEP, and I would like to stress the point that it's not PEP. It's actually very, very different. And I would like to stress some of the clinical features that distinguish SJIA lung disease from PEP, and then Grant will talk a little bit about the pathophysiology. So coming back to this picture that I showed you earlier, right? First of all, we see this massive interstitial infiltration. If you look at sort of classic PEP, right, we have this significant filling of the alveolar space with this proteinaceous material, but if you look at the interstitial space, it actually looks pretty decent. And correct me if I'm wrong, in PAP patients, you may see fibrosis and alveolar remodeling, but it usually happens 10, 15, 20 years from the, so usually, certainly, it's not a feature at the early stages of the disease. And coming back to our patients, I mean, look at this massive inflammatory activity that leads to a massive structural alveolar remodeling. So, you know, as any of you who is not a pathologist, would you recognize that this is a lung? So that, again, that's something that is very, very different from PAP. We do not see this in PAP. And I think probably the same true for fibrosis as well, correct? So you may see a little bit, but again, it's sort of a very, very, very late finding, and that we see these kind of things relatively early in our patients. So my pathologist is telling me if that sort of, if it's kind of yellowish staining, that it's pretty irreversible, right? Sort of if it more kind of greenish, bluish, so some of it still may be reversible, but anyway, the point is whether reversible or not shows up very, very early in the disease, and that is strikingly different again from patients with PAP. And now I will ask Grant to chat a little bit about the pathophysiologic differences between PAP and the disease that we are seeing here.
Can you take like 7 minutes?
Yes, I can do exactly 7 minutes. I think we're gonna try to power through, and then, or do you wanna, we can have one question.
I'm just curious because GATA2 haploinsufficiency also tells us PAP and have varying degree of interstitial findings too. Like when you're talking about classical PAP as a GM-CSF receptor defect or a surfactant production problem, I think a better control to compare this type of disease would be a SAMD9 and IRF8 and GATA2 all have varying degree of pulmonary findings that mimic secondary PAP.
I agree. Actually, that Chris Tao, the pulmonologist we work mostly with, actually says GATA2 probably looks the most similar to the pathology of this disease. I agree with that. So I'm gonna, 5 minutes now.
Can't get some. Do GATA2 mice or patients have high level of CD4 in the lungs?
There are
Type-
GATA2
type TH1.
haploinsufficient mice, but none of them develop, as of I know, it's mainly a sort of hematopoietic phenotype. Biallelic GATA2 is embryonically lethal,
Okay.
so only haploinsufficient mice, you need to challenge them really bad with inflammatory phenotype to show some hematopoietic stress there. Otherwise, the mice itself is not awesome model to show lung pathology.
All right, so I'm going to just try to make a couple of points, and I think particularly ones that kind of complement and build off of things Raffaella was talking about. So these first couple slides are from our published paper. So this is looking at the cytokine profile of the BAL from Illuminex that we use. Again, we see significantly elevated IL-18 in the BAL from these patients. As Raffaella said, variable expression of CXCL9 as well as 10, very high in some, but certainly not all. Basically, all of the other cytokines, IL-6, IL-8, TNF, several others, were really quite low and really indistinguishable from sort of primary PAP patients that we used as a comparator. We didn't look specifically for Th2 cytokines like IL-4 or IL-13, which I think would be interesting to think about. So this is also some gene expression data. So this is a nanostring panel that we used, which looks at about 750 genes related to inflammation and autoimmune pathways. And this was done on a whole lung tissue from the biopsies. So the nanostring platform, because of the technology, is able to get good, very reproducible data from degraded RNA such that you would see in formalin-fixed paraffin-embedded tissues. So we did a sort of a pilot experiment looking at eight samples from biopsies from 5 different patients with this disease. We compared it to 3 healthy lungs that were from our lung transplant program that were used as controls. And what you could see is we, after correcting for comparisons, we found about 26 or 27 genes that were significantly altered, most of them being upregulated. And then a couple of, again, you can't, because it's not an unbiased survey, you can't really do a true pathway analysis, but you can sort of look at patterns. And we saw a couple things. One were a number of genes that were annotated for being in type 2 interferon pathways, although I would note that most of these are also in type 1 interferon pathways. So it's, as Raphael said, it's very difficult to disentangle those. But you can see CXCL10, CXCL9, as well as several other ones. There are also several that are related to T-cell related genes, CTLA4, the IL-2 receptor. Interestingly, not really genes that were linked very strongly to a Th1 kind of response that we saw in this, at least after correcting for comparisons, which again is a high hurdle for a small number of samples. I'm sorry, did I say Th1? I meant Th2. And then this is just another way to look at some of the data. This is the type 2 interferon signaling pathway. The red genes were those that were upregulated. This is the broader universe of those with a lower threshold for significance. It looks a little bit more overrepresented because of STAT1, which is sort of all the way through this, which is upregulated. But STAT1, as well as a number of STAT1 related genes, were also upregulated in this. So again, I think there's strong evidence for type 2, certainly an interferon broadly, whether it's type 1 or type 2, I agree. I think that's a really difficult question to get at. I think another important point from this was that we saw very similar signatures in affected versus unaffected areas of the lungs. So for 2 patients, we had biopsies from multiple locations of the lung. So the surgeon, Dr. Dasgupta, tries to get an area that looks very involved and an area that looks grossly uninvolved. Those grossly uninvolved areas have a little bit of disease. So this one here, there was actually sort of hemorrhage. There's a lot of red cells, so it looks busier than it really is. This one, it's a little bit cleaner. So you can see it's not 100% clean, but it's much better versus these areas that are much more extensively involved. This is looking at the principal component analysis for the nanostring. Really, the unaffected areas cluster exactly with the affected areas. So we don't really see any major differences, which is interesting and suggests that maybe a lot of the signature is not being driven as much by the inflammatory cells and potentially more the lung parenchymal cells. But I don't know. And then the other thing that this sort of raises with this sort of strong interferon gamma signature that we see is some work from a couple years ago that showed that when Th1 is being sort of constitutively overexpressed by one of its main drivers called TBET, you get a phenotype in mice that looks very similar to this. So you get this really significant dysfunction of bone marrow macrophages, getting hemophagocytosis and erythrophagocytosis, which could look a lot like HLH-MAS. But you also see dysfunction of the alveolar macrophages with development of a PAP-like lung disease. So it suggests that maybe a constitutive activation of this pathway could lead to pulmonary macrophage dysfunction. And this is from their paper. So again, I think we see some overlapping pathophysiological features in SJIA lung disease and MAS, particularly this gamma interferon, as well as high IL-18, suggesting that there could be a link between these, which we see clinically as well. And so this is sort of the sketch and a model from our paper, and I always make the joke that when you see a model with this many arrows, you know it's wrong, because a real model isn't going to be this complicated. But trying to understand that potentially there's this cytokine storm milieu that you see in MAS, including high IL-18, including activated interferon gamma, where you see activation of lung macrophages, a Th1-driven response, production of these interferon-induced signature, but then potentially dysfunction of the alveolar macrophages that leads to defects in surfactant recycling and then the clinical phenotype that we see. Again, lots of question marks. What are the biologics doing? Are there other things going on? Are there genetic factors? I want to very quickly show, and Alex may show a little bit more of this data tomorrow, but just because it gets back to a point that you made, which I think is super interesting. So we've done one experiment on doing a single-cell RNA-seq experiment on a lung biopsy from one of these patients. We had some technical issues. We didn't get nearly as many cells as we were hoping to, but it's enough to kind of see some different populations. So when you look at those 800 or so good cells we can see clear differences in identifying multiple different cell types here. And if you kind of break them apart and try to annotate what there are, so what we saw were what we were hoping to see, which is both parenchymal cells as well as inflammatory cells. So we see epithelial cells, both ciliated and non-ciliated. We see endothelial cells. We see T cells, both CD4 and CD8. We see some NK cells. We see a couple different macrophage populations. One looks more like recent emigrants to the tissue. Or no, this is whole lung. This is tissue. This is tissue. But then we saw this other population that when our first team took a pass at it, they just called it cluster 11 because they weren't clear what it was. Alex looked at it and thought it looked somewhat mast cell-like. And I thought I put another slide in there on this, but I think that it's mast cells, which is very interesting thinking about what Raffaella was presenting and potentially the role for those sort of similar cytokines driving mast cell activation in such this disease. Correct. So we also tried to do the same experiment in that unaffected area of the lung. And there were some technical issues with that also, but we also, we saw the parenchymal populations. We didn't see nearly as many of these inflammatory populations, and we didn't see the mast cell population in the unaffected area. So, so interesting that there may be some evidence of mast cells in the lungs of these kids as well. So I think I'm going to leave it at that.
You didn't see any dendritic cells?
We didn't, but our sequencing depth again was suboptimal. So we were hoping for 5,000 to 7,000 cells, and we got about 800. And I'm not entirely sure why. We're working on that.